Welcome to this lecture on the principles of immunology. Today, we are going to explore one of the most fascinating and crucial systems in our bodies, the major histocompatibility complex, or MHC. If you have ever wondered how your body knows exactly which cells are yours and which belong to a dangerous invader, you are in the right place. Let us dive in and uncover how this microscopic identification system keeps us alive. To start, imagine you are a security guard at a highly secure facility. Your job is to check identification badges. In your body, T cells act as these diligent security guards. However, they have a major limitation. They are completely blind to what is hiding inside a cell. To solve this, our cells use a special display window, which is the MHC. By constantly presenting little pieces of internal proteins on these MHC molecules, the cell proves it is healthy or raises an alarm if infected. Now that we understand the analogy, let us look at the formal biological definition. The major histocompatibility complex is a tightly linked cluster of genes. These genes encode highly specialized cell surface glycoproteins. Their fundamental job is to bind to small peptide fragments derived from pathogens and display them on the cell surface. This presentation is what allows the appropriate T cells to recognize the threat and mount an immune response. Building on that definition, there are two primary classes of these molecules that you need to understand deeply. First, we have MHC class 1. These are found on almost every nucleated cell in your body and specialize in presenting endogenous antigens, like proteins from a replicating virus inside the cell. On the other hand, MHC class 2 molecules are exclusively found on professional antigen-presenting cells, like macrophages and B cells. They present exogenous antigens that have been engulfed from the outside environment. You might be wondering, why does this complex presentation system matter so much in real life? Without MHC molecules, our adaptive immune system would be paralyzed. T cells cannot recognize free-floating antigens. They only see them when properly seated in an MHC molecule. This is known as MHC restriction. Furthermore, because every person has a highly unique set of MHC molecules, a transplanted organ from a stranger looks like a massive foreign invader. This is why finding a match for bone marrow or organ transplants is so difficult. This brings us to one of the most fascinating aspects of the MHC, its incredible genetic diversity. This diversity ensures that a population can survive new and evolving diseases. It comes from two main sources. First is polygyny, which means we have multiple different genes for MHC. Second is polymorphism, meaning there are many different versions or alleles of each gene in the population. Together, these create unique genetic combinations known as haplotypes. To truly grasp this diversity, we can represent it mathematically. The total number of possible unique MHC combinations in a population is found using a simple combinatorial multiplication formula. N equals A1 times A2 times A3, and so on. Here, N represents the total number of unique genetic combinations. The variables A1, A2, and A3 represent the number of known alleles for each specific MHC gene locus. If the number of alleles for any single gene increases, the total combinations increase exponentially. While the population has millions of combinations, an individual has a limited set. Let us look at the formula for calculating how many unique MHC class 1 molecules a single fully heterozygous person can express. E equals G times 2. In this formula, E represents the total number of unique MHC molecules expressed on the cell surface. G represents the number of gene loci. We multiply by 2 because humans are diploid, meaning we inherit one allele from our mother and one from our father, and both are expressed simultaneously. Let us apply this formula with a concrete example. We want to find out how many unique MHC class 1 molecules are present on the cells of a completely heterozygous human. We know that humans have 3 main MHC class 1 genes, HLA-A, HLA-B, and HLA-C. So our given number of gene loci, G, is 3. We substitute this into our formula, giving us E equals 3 times 2. Solving this gives us a final answer of 6. This means every nucleated cell in this person will express exactly 6 different types of MHC class 1 display windows. Now that we have calculated individual expression, let us explore how these genes are passed down, which is critical for organ transplants. MHC genes are located very close together on the same chromosome. Because of this, they are inherited as a complete block, which we call a haplotype. A child will inherit one complete haplotype from their father and one complete haplotype from their mother. This Mendelian inheritance pattern allows us to calculate the probability of 2 siblings being an exact genetic match. To find the chance of a perfect match between siblings, we use a basic probability formula. PT equals P1 times P2. Here, PT is the total probability that 2 siblings share the exact same genetic makeup for their MHC molecules. P1 is the probability of inheriting the same haplotype from the first parent, and P2 is the probability of inheriting the same haplotype from the second parent. Since each parent has 2 haplotypes, the chance of the second sibling getting the same one as the first sibling is 0.5 for each parent. Let us walk through this real-world example. A patient needs a bone marrow transplant. The father has haplotypes A and B, and the mother has C and D. The probability that a sibling inherits the exact same haplotype from the father is 0.5. The probability of inheriting the exact same haplotype from the mother is also 0.5. We multiply these individual probabilities together, 0.5 times 0.5. This gives us a final answer of 0.25. This means there is exactly a 1 in 4, or 25% chance that any given sibling will be a perfect MHC match. Having done the math, let us visually examine the physical structures of these molecules. On the left of this diagram, you see MHC class 1. Notice how it consists of a single large alpha chain paired with a smaller protein called beta-2 microglobulin. This creates a closed-ended groove at the top that holds short peptides. In contrast, on the right, MHC class 2 is made of 2 roughly equal-sized chains, an alpha and a beta chain. This forms an open-ended groove that can accommodate much longer peptide fragments. These structural differences dictate how they function in the immune system. We have covered a lot of ground today, so let us summarize the most important ideas. First, remember that the major histocompatibility complex acts as a crucial display window, allowing T cells to inspect internal cell proteins. Second, MHC class 1 handles endogenous threats from inside the cell, while class 2 handles exogenous threats from outside. Finally, The incredible genetic diversity of these molecules protects our population, but makes organ transplants challenging, with siblings having only a 25% chance of a perfect match. You now have a solid understanding of the structure and functions of MHC.